Today we're going to be learning about the cell cycle. All cells go through a life cycle just like people do, just like any living organism does. But on the cellular level, they also go through a cycle of life. So we're going to represent that right here as a circle. Alright, now as you can see there are different sections of this circle and we're about to go over every single step of this cycle. So the first step is this long black line that we're going to call interphase. Interphase is a time when the cell performs its normal job. It does what it was made to do. But meanwhile, in the background, it is going through different processes to prepare it to split off into two different cells. So, let's consider what a cell has to do to prepare to divide. So, let's say we have a cell here, a little worker cell, that is ready to divide. But instead of going through the processes that we're about to discuss, he instead just decides to split. And so he splits. The problem here is that each of the new cells is at a disadvantage. They only have half of what they need to survive. They only have one eye each, half a smile, even half a hard hat. So what does it have to do to prepare for this? Well, logically, it would have to double everything. If you want to have two equal daughter cells, you have to double everything. That's DNA, organelles, mitochondrion. So for this, he has 4 eyes, 2 mouths, 2 hats. And so now he's able to make 2 equal cells. So what makes up the interphase? Well, the first phase is called G1. While the cell is performing its normal job, G1 is occurring, which means the cell is replicating all its contents, but not the DNA. All the proteins and other organelles are being doubled, but remember, DNA is not being doubled. The second phase, right here, in this corner, is the S phase. S stands for synthesis, and as we've learned earlier, synthesis usually is correlated with DNA. DNA synthesis, which is when the DNA is doubled. So, you have G1 phase, where some of the cell contents are beginning to be doubled. You also then have the S phase, where the DNA is doubled. And then you finally have G2. The G2 phase, we're not going to get into too much of what's going on during this, but I just want you to remember that this is where the cell prepares and grows so that it can split into 2 even normal-sized cells. So we've gotten to where the cell has duplicated everything that needs to be duplicated, and the cytoplasm is big enough to where it can divide into 2 different equal cells. This stage of division is called mitosis, represented by the red arrow. So during mitosis, there are 5 different phases that I'm going to walk you through. So first, it's called prophase. Prophase is where the cell condenses its DNA, as you can see, within the nucleus, which actually, while it condenses the DNA, the nuclear envelope starts to disintegrate. And while that is happening, you have these things called centrioles that start making microtubule fibers. These fibers are going to be used to split the DNA later in this process. Then we have metaphase. Metaphase is where the microtubules have attached to the center of each chromosome, or sister chromatids, at the centromere, and lines them up evenly down the middle of the cell. Now, I want you to note that these X's of chromosomes, these are the replicated sister chromatids, as in 1/2 of this is the full amount of DNA. In fact, this is actually the doubled amount of DNA. Let's say that since there are 6 sides, that really all a cell needs is 3. But instead, it's duplicated it because of the S phase, synthesis, so that when it does divide, 3 goes to one side and 3 goes to the other. That is metaphase, where it lines up attached to the centromere by the centrioles spindle fibers. The next phase is where they split, anaphase. Now, anaphase is obviously, the next stage that's going to happen. If you line them up in the middle, you got to divide them. And so what happens is that the chromosomes, the sister chromatids, detach at the middle and are pulled by the centrioles into their opposite poles, to the opposite sides. And now, the final stages of mitosis is really divided into two parts. So the first one is telophase. Telophase is obviously where you have the 3 chromosomes from going to either side. In this particular cell, we all know that our cells have many more chromosomes. For the simplicity, I made it just 3 per cell. You see that now that they have gone to their opposite sides, they have started to create their own nuclei. The splitting of the cytoplasm is called cytokinesis. Cyto as in cytoplasm, kinesis as in the breaking apart. And so after all of these, we have gone from one cell that has had all its DNA and things that needed to be replicated, went from one of those into 2 equal, identical cells. And that is mitosis. So what I've gone over is the basics of what the cell cycle is. There are many more details, such as checkpoints, the G0 phase, that we're going to go over in future videos. But for now, this is the basics of the cell cycle and what you need to know. For class tomorrow, you must be able to take a quiz on this, telling me what happens in each phase of mitosis and interphase. If you do have any questions, please do not hesitate to ask me in tomorrow's class.